Thanks everybody for coming. Hello, my name is Kevin Collins. I'm in the biology department at Coral Gables campus. I'd like to welcome you all to the neuroscience seminar today. It's my pleasure to introduce Dr. Lucas Cheadle from Cold Spring Harbor as our speaker today. So just a little bit of background on Dr. Cheadle. So he got his undergraduate degree from Smith College and then went to Yale University to pursue a PhD in neuroscience, which is actually where I got to know Lucas. So Lucas was a graduate student in Tomas Peter's lab, where he studied kind of the mechanisms of synapse assembly. And he was in the floor above me in the Department of Biophysics and Molecular Biophysics and Biochemistry department, which was a very fun department to do neuroscience in, I should say. This is the department where Joan Stites and a bunch of RNA structural biology people were there. We were among the 3 labs, I think, in the whole department who studied questions in neuroscience. So it was a very fun department to be in and try to sell people on the importance of neuroscience research to a bunch of biochemists and biophysicists. So after that, Lucas then procured a very highly coveted postdoc position in Michael Greenberg's lab at Harvard Medical School, where he completed his postdoctoral work. So Dr. Lucas has done some fantastic work kind of studying synapse assembly, the mechanisms of synapse assembly, and then as a postdoc kind of introduced the idea of studying activity-dependent plasticity as well, that remodeling a circuit function. And so he's been highly regarded and he's gotten great support for his research since joining Cold Spring Harbor. So before that, he got a K99 award, which he's now transitioned into R00 award. Since going to Cold Spring Harbor, he's been a recipient of the Brain and Behavior Research Foundation Young Investigator Grant, a Rita Allen Scholar Award, the McKnight Scholar Award, many of these like happening just like within the last few months or something like that, I think. And basically just hopefully plowing his way into high success. That's our goal here, is to kind of see how far Lucas can take this field, which I think is a very interesting kind of a combination of cell biology and some neural circuitry and some behavior and putting it all together. So it's my pleasure to welcome Lucas for his seminar today, which is entitled Microglia and Fn14 Regulate Neuronal Transcription and Chromatin to Shape Circuit Refinement and Function. Thank you, Lucas.
So thank you so much for that kind introduction, Kevin. It was really great to hear from you. When I got your email, I recognized your name immediately, and I thought, oh, this is the postdoc that was always running around Michael Cole's lab and being really productive. So it was nice to hear from you. So I appreciate the invitation to speak to you guys today. This is some work that we've been doing in my lab at Cold Spring Harbor, where I just started about a little over a year ago. And to some extent, it's following up on a discovery that I made as a postdoc in the Greenberg lab of a particular cytokine pathway and how it plays a role in circuit development. So the work that I'm going to talk about is something that we're actually trying to get submitted over the next week. So it's a really nice time point to get some feedback on it. So a question that's always really fascinated me is how the brain transforms from a handful of undifferentiated progenitor cells into one of the most complex networks in existence. And as you might imagine, this process is incredibly multifaceted, and it proceeds through a number of different developmental stages, which are both temporally segregated as well as overlapping. And in the human brain, development has sort of evolved to be an extremely protracted process that begins in the 3rd week of gestation and extends into the 3rd decade of life. But in the mouse, which is the model studied, brain development is actually accelerated such that it's largely complete by about 1 month after birth. And that makes it a really tractable model for asking questions about the mechanisms guiding neural circuit development and function. And so I think what most people are aware of is that the connectivity of the brain really is established through both intrinsic genetic mechanisms as well as environmental cues. And for example, we know that the earliest stages of neuronal differentiation and synaptic assembly can largely occur through intrinsic molecular and genetic mechanisms. And that includes things like transcription factors, which specify the core identities of neurons, to molecular guidance cues and ligand receptor interactions between post and pre-synaptic neurons that allow them to sort of assemble these nascent connections. And actually, these intrinsic mechanisms are really sufficient to establish a rough map of synaptic connectivity in the brain by about birth. But this synaptic connectivity map that's established through these mechanisms is actually far from mature. Actually, these immature connections need to then go through a further process, which we call synaptic refinement. And this process is really involves the selective strengthening and maintenance of a subset of those synapses that are initially formed, and the concurrent elimination of many of those synapses that end up not to be super important and therefore get removed. And while the earliest stages of synapse formation appear to be largely driven by intrinsic mechanisms, this process of synaptic refinement is really sensitive to the influence of sensory experience or information from the environment. And so we really know very little about the basic mechanisms that underlie this process of refinement. And so this has really become a key interest in my laboratory. So the visual system has been for a very long time and continues to be a really useful experimental system for understanding sensory experience dependent refinement in the brain. And something that's great about the visual system is that a lot of the principles that have been discovered that are really fundamental to visual system development can actually extend to other circuits as well. So it's a way to really investigate some of the sort of generalizable features that characterize postnatal brain development. And in particular, my lab takes advantage of a circuit which is called the retinogeniculate circuit, which is the connection between the axons of retinal ganglion cells in the eye, which I'm showing here in orange, and their postsynaptic targets in a small brain structure called the dorsolateral geniculate nucleus, which I'm showing here in teal, and I'll call that the DLGN throughout the talk, which resides in a deep brain structure called the thalamus. And so the DLGN receives input from the retina, and the neurons in the DLGN, which are called thalamocortical or relay neurons, and I'll kind of use those terms interchangeably to some extent. Once they receive input from the retina, they project information to the visual cortex, which is the higher order visual processing center of the brain. And you can actually visualize this small circuit and structure in the brain by using, for example, a lipophilic tracer, which you can inject into the eye to trace the retinal axons. And you can see here in red that those axons are labeling also their terminals in the DLGN. So it's a really important brain structure, and the connection between the eye and the DLGN is a fantastic model for studying experience-dependent refinement. And the reason why it's such a fantastic model is that refinement in the DLGN has been extensively characterized at the physiological level across the first month of life. And I'm going to just schematize what that process looks like here instead of getting into the nuts and bolts of the assay. So here I'm showing you a given thalamocortical relay neuron in the LGN as an oval that's teal, and I'm expressing sort of to visualize retinal ganglion cells contacting that neuron in orange. And so each of these orange circles is one retinal ganglion cell converging on the relay neuron, whereas the weaker inputs are displayed by smaller circles, and the stronger inputs are displayed by bigger circles. So what I hope you can appreciate is that around the first week of life, prior to eye opening, which occurs around P14 in the mouse, each relay neuron in the DLGN receives relatively weak input from a large number of retinal ganglion cells. But as the brain matures and the eyes become opened up, the number of retinal ganglion neurons that converge onto that cell decreases while the strength of the connections that remain increases. And finally, by the time you get to about P30, each thalamocortical neuron receives relatively strong input from only a small number of retinal ganglion cells. And we know that this precise convergence property of the DLGN is really important for its function. And so an investigator named Dr. Chin-Fei Chen, who is a good colleague of ours at Boston Children's Hospital, had actually asked whether this process requires sensory experience by placing mice in the dark, so complete darkness for the mice, during different periods of this process. And what she found was that visual experience is really dispensable for refinement up until about P20, but between P20 and P30, experience is critical for the further refinement of the circuit. And so this really established, in my mind, a nice experimental platform that could serve as a good jumping off point to start to understand the cellular and molecular basis of this process of experience-dependent refinement. So as a postdoc, I took advantage of this model, and I used a stimulation paradigm in which we rear mice according to a standard light-dark cycle here, normally until P20, but we then put them in complete darkness between P20 and P27, which is really about the peak of experience-dependent refinement in the mouse. We can then take the brain and we can dissect it in the dark so that we have our sample that's completely unstimulated, or we can re-expose the mice to light for about 8 hours and then dissect out the DLGN. And we know that that paradigm does a great job of inducing neuronal activity in the DLGN, which leads to the expression of activity markers like phos, as well as remodeling of circuits. So it's a nice discovery tool to start to understand mechanisms underlying refinement. Now, just to briefly summarize what we discovered through this assay is performing single cell sequencing on the DLGNs from dark reader stimulated mice allowed us to first identify in our data set the thalamocortical neurons, that's what I'm showing here in gold, and next to ask whether there were transcripts that were significantly more or less expressed in the stimulated cells versus unstimulated. And the bottom line is that we did identify a number of genes that were either up or down regulated with stimulation. The single most highly induced gene that we observed was a gene called Fn14. Fn14 stands for fibroblast growth factor inducible protein of 14 kilodaltons. And it's actually a member of the tumor necrosis factor receptor superfamily. So it's a family member of the TNF alpha receptor, you can think of it. And this is a relatively large family of pro-inflammatory cytokines outside of the brain. Fn14 happens to actually be the smallest member of the family. So it's a transmembrane protein, but it only has a very tiny cytosolic tail. And so I was very intrigued to find this sort of pro-inflammatory receptor being transcriptionally responsive to visual stimulation. And so I sort of started thinking about what it might be doing in the context of the brain. And looking at what its function is outside of the brain really painted a picture of a molecule that in healthy tissues is typically quite lowly expressed, but becomes significantly upregulated in response to injury or pathology. And upon that upregulation, it becomes sensitive to Tweak, which is its ligand. It's a cytokine of the TNF family that binds Fn14, and it's expressed in infiltrating macrophages. So when you have a site of injury, you turn up the Fn14 expression, you now have macrophages coming in and they're releasing Tweak. So now you have Tweak Fn14 signaling and it leads to tissue remodeling, for example, fibrosis. And so in my mind, this is very interesting because, tissue remodeling and circuit remodeling, they're kind of analogous in a lot of ways. And so we essentially followed up on this by taking advantage of an Fn14 knockout mouse line and asking what effect knockout of Fn14 has on synaptic refinement. So just to summarize the result of that experiment, because this work has been published, what we observed is that refinement occurs normally in the absence of Fn14 all the way up to about P20. But specifically during that phase of experience-dependent refinement, loss of Fn14 essentially blocks the further refinement of the circuit, both at the level of synaptic strengthening as well as at the level of synapse elimination. And consistent with this role for Fn14 being specifically linked to sensory experience, the phenotype of the Fn14 knockout mice looks very similar to what we would see if we simply put the mice in the dark. And so our next question was, if Fn14 is important for this process, what about its canonical ligand? So we wanted to know where this ligand Tweak was expressed. And so we looked in a single cell data set that we had recently published, and we wanted to know where Tweak was during postnatal development in the DLGN. And what we observed is that prior to eye opening, Tweak is relatively lowly and ubiquitously expressed across a number of cell types. But right around the time of eye opening, Tweak expression becomes significantly upregulated, specifically in a class of cells called microglia, and its expression continues to be increased in microglia into the phase of experience-dependent plasticity and refinement in the DLGN. So that was really an exciting result for us because microglia are a very interesting type of cell. They are essentially the most prominent type of non-neuronal cell in the brain that is not derived from a neural lineage. So microglia are actually derived from myeloid progenitors in the yolk sac. They actually develop and colonize the brain embryonically prior to the formation of the blood-brain barrier. So that's how they make their way into the brain. And they essentially tile the brain, and they play an important role in the brain's response to injury and pathology. But work over the past decade or so has begun to uncover really critical roles for microglia in healthy brain functions as well, probably the most prominent of those functions being to regulate synaptic refinement by pruning synapses during development through phagocytosis. So phagocytosis provides a method through which microglia, particularly in the LGN during the presensory phases of development, is actually able to engage cytokine signaling to essentially eat synapses, which removes those excess synapses that, remember, removal of them is an important process of normal brain development. And so we were able to come in with studies of Tweak and Fn14 and actually show that the roles for microglia in synaptic refinement extend into experience-dependent phases that was not known before. But additionally, that what happens during the experience-dependent phase is not phagocytosis. So it's a completely different mechanism. And essentially, as I'm schematizing here, the way it works is that microglia release Tweak, it binds to Fn14 at synapses, and this leads to the disassembly of synapses. And so emerging from my postdoctoral work and coming to Cold Spring Harbor, one of the things I was really interested in doing is taking advantage of this pathway as sort of a molecular handle to try to understand the ways that microglia can impact neuronal development and function that are distinct from these phagocytic pathways that are really the topic of, I would probably say, the majority of microglial work that's being done in neuroscience around this time. And I think that it's really important to study that, but I think it's, for me, a little bit more interesting to think about the idea that microglia could be doing many more things that may not even involve the process of phagocytosis in the to begin with. And so we wanted to understand this mechanism of Tweak Fn14 synapse remodeling. And we already had a little bit of a hint that this might be occurring through sort of a unique mechanism beyond just sort of the disassembly of individual synapses, because Tweak and Fn14 outside of the brain actually mediate a lot of processes through transcriptional control. And so, for example, this review article really nicely describes some of the downstream pathways that are induced by Tweak Fn14 signaling, and that can either occur through Tweak Fn14 binding, or Fn14 can also signal on its own. And that these events can actually drive, for example, the activation of the MAPK pathway, the activation of the PI3K pathway, and what's been best described is the activation of the NF-kappa B transcriptional complex. NF-kappa B is a really classical, really important transcriptional mechanism, particularly in immune cells, really important for immune function. And so it's known that Fn14 can sort of regulate that process outside of the brain. So I thought it would be very interesting if perhaps Tweak and Fn14, rather than simply regulating synapses on a synapse to synapse level, maybe playing a broader role in shaping the transcriptomic profile of neurons as the brain develops. And so the work I'm going to show you now is the study that we're currently trying to get submitted. And this is work that's been really led by a postdoctoral fellow in my group, Austin Farrow, who's been very talented, and he's only been here for about a year. So it's really incredible the amount of work he's been able to do. So we decided that if Fn14 is important for regulating gene expression in thalamocortical neurons, then if we actually looked at gene expression in the knockout mouse, we could identify some of the Fn14 target genes. And so that's what we did. We took the DLGNs of P27 Fn14 knockout and wild type littermates, which, as a reminder, I'm showing here with this RNA scope experiment that Fn14 is specific to the thalamocortical relay neurons in the DLGN. I'm showing that with a co-stain of Fn14 in green and VGLUT1 in magenta. And so we did single nucleus sequencing on these DLGNs right at P27 at the height of Fn14 dependent, experience dependent refinement. And so this experiment allowed us to take a look specifically at the different types of cells in the DLGN, as I'm showing here on this UMAP, which is a two-dimensional representation of single cell data. I'm sure many people are used to seeing these by now. It's become a really important technique in the field of neuroscience, but essentially the map is simply a way to represent the heterogeneity in the system such that each data point represents a cell in the data set. And its proximity to other cells really displays the heterogeneity or the transcriptional divergence. So cells close together are similar transcriptionally and cells far different. And we were able to selectively look at the thalamocortical neurons in the data set because we could see that an important marker of these cells, PRKCD, was really enriched in these clusters up here. And there were 4 clusters of TC neurons we pulled out. But we know from our work and from the work of others now that TC neurons, even though they may cluster slightly differently, transcriptionally, they're all very similar. So we really focused the bulk of our analysis on the 2 largest TC neuron clusters on the basis of that. And what we think is really interesting here is that the genes that we found to be misregulated in the absence of Fn14 were largely misregulated in these TC neuron clusters, which makes sense because those are the cells that express Fn14. And so in this bar graph, I'm simply showing you sort of the counts of the numbers that were either significantly downregulated in the knockout or upregulated in the knockout. So while the most genetic transcriptional changes occurred in TC neurons, I also want to point out that we found quite a few changes in an inhibitory neuronal subtype as well. But when we looked back at that subtype in tissue, we noticed that that was actually a population of cells that are very close to the DLGN. And so we inadvertently captured a few of those in our dissection. So essentially, this leads us to conclude that the vast majority of misregulated genes are found in the TC neurons and likely represent some role for Fn14 in mediating transcription, rather than simply seeing differences in gene expression because the circuit itself is less mature. And when we look at the functions of the genes that seem to be misregulated in the Fn14 knockout, their functions make a lot of sense in terms of what we know about their roles in refinement. So for example, genes that are downregulated in the knockout tended to encode regulators of synaptic organization, glutamate receptor signaling, dendritic spine morphogenesis, processes that are really crucial to synaptic remodeling, which is something that we know Fn14 does. We also found genes to be upregulated in the Fn14 knockout, consistent with the ability of Fn14 to both drive and repress transcription in the periphery. And we noticed that a lot of the upregulated genes also had to do with synaptic function, for example, neurotransmitter levels, synaptic signaling. And we think that also makes sense because we think it's likely that Fn14 is probably sort of upregulating some synaptic genes, downregulating others, potentially to sort of shift the profile of these synapses towards favoring disassembly, for example. But what we thought was particularly interesting in these data was that we also identified a number of mediators of chromatin remodeling as well as histone modification, specifically in the gene sets that are downregulated in the Fn14 knockout. And we didn't necessarily expect to see that. We were more thinking that we would see a lot of synaptic genes, not so many regulators of transcription in and of itself. And so, of course, one of the first things we did was try to validate some of these transcriptional changes that we found by single-cell-seq, and we were able to do that by qPCR. I'm just showing a few examples here. So it does look like these are pretty robust changes for the most part. So why are we excited about the possibility that Fn14 could go beyond regulating synapses to actually regulating what's happening in the neuronal nucleus? And it's because those events that happen in the nucleus are super important for the epigenomic maturation of neurons, which we know is occurring sort of concurrently with experience-dependent refinement. And so I don't know everybody's background, so just a really brief description of what these chromatin remodelers and histone modifiers do. So genomic DNA in the nucleus does not sort of exist in a naked state. Typically, it is sort of tightly wound around protein complexes that we call histones. And these histones have these tails on them that you can actually add or subtract different types of post-translational modifications to. And there's a large number of different modifications, but a couple of the most prominent ones are methylation and acetylation. And what these modifications are capable of doing is actually moving that histone around so that you can actually make genes that were previously occluded. So in this example, we have a gene that can't be transcribed because it's all sort of occluded by all this histone, versus whenever it's in more of an open state and it's capable of being expressed, because now transcription factors combined, regulatory machinery combined, and it can become open. So what's interesting about these histone modifiers is that by modifying these histones, they can actually determine which parts of the genome are open and ready to be transcribed, which parts of the genome are closed and can't be. So the histone modifiers that we found to be particularly downregulated in the absence of Fn14 fell mainly within a couple of groups. So we found that HDACs, or histone deacetylases, were downregulated in the knockout. But we also found that a very large number, relatively large number of histone lysine demethylases tended to be downregulated in the knockout as well. And you can tell from these fold change differences that we're not talking about really massive changes. But I think what's interesting here is that just the pattern that there's a host of these molecules downregulated overall. So I think sort of small changes in these molecules, when you add them up, could lead to, you know, Fn14 being able to potentially play an important role in shaping the chromatin landscape in the neuron. And so we next asked whether loss of Fn14 would change the chromatin landscape. In other words, the accessibility of different parts of the genome to regulate transcription or to be transcribed. And so to answer that question, we used an approach called assay for transposase accessible chromatin, followed by sequencing. And this is a way that we can, in a genome-wide manner, measure or basically profile the accessibility of chromatin across the genome. So briefly, the way it works is you essentially take your DNA, where again, you'll have some sites that are closed and you know, the genes are not made available, or then you'll have open sites as well. And you actually put in sort of a modified enzyme that's capable of cutting in open regions of chromatin. So it can't cut where the chromatin's closed, but it can where the chromatin's open. And you can use that enzyme to actually integrate adapters, sequencing adapters, into those spots where the chromatin is available. And what that means is you can then fragment those and you can amplify them, do a library prep and sequence them, and then you can map where are these adapter-associated regions across the genome. And wherever those adapters are at, it means that those regions of the genome were accessible for binding of transcriptional regulators. And so we pulled some tissue from the DLGNs, the cortex and the hippocampus of Fn14 knockout wild-type mice. And then we performed attack sequencing. And so just to orient you to what these traces look like, I'm going to show a few traces in the course of the talk that look like this. And essentially what I'm showing here is on the x-axis, a genomic locus. In this case, I'm showing you the Fn14 locus. And on the y-axis, I'm sort of showing you accumulations of attack sequencing reads. So we call these peaks, and the higher the peak, the more accessible the region is. So I think this is a good one to start with because it's a validation of the approach, because you can actually see a lot of accessibility here in the wild type around the Fn14 locus, but that's essentially ablated in the knockout because it doesn't have Fn14. So it's a nice control. Additionally, even though we did pool cells from this mouse, so we're not only getting neurons in the data set, by looking at a couple of other databases where you can put in your differentially accessible peaks and have it predict what types of cells you're looking at, it does look pretty consistent that a lot of what we're seeing is neuronal. So we actually found quite a few peaks that were either more or less accessible in the Fn14 knockout or wild type neurons. And we specifically identified 197 peaks that had less accessibility in the knockout and 503 peaks that had more accessibility in the knockout. And I'm just showing those plots here, as well as a volcano plot. So this is really interesting because it tells us that there's this robust change in the chromatin profile of the genome when you lose Fn14. Here again, I'm just showing you a couple of these example sites. So in this case, we're looking at a gene called DLGAP2, which is an important excitatory postsynaptic scaffolding molecule. As you can see, this particular intronic, it's in an intron, this particular intronic peak is more accessible in the knockout than the wild type. And we can see the opposite effect if we look at this intron for a gene called LINGO2, which has been associated with Parkinson's disease. So we see pretty robust changes in chromatin accessibility in this context. And we were next interested in understanding what are the functions of the genes that might be regulated by these cis-regulatory elements. And so we annotated the genes that we would think that these peaks might be regulating. Of course, we can't know for sure, but there are ways to estimate this, particularly, for example, the proximity of a gene body to a regulatory element. But at the end of the day, we could perform gene ontology analysis to look at what functions these regulatory regions might have. And again, we see that there are a number of categories such as transsynaptic signaling, chemical synaptic transmission, some of the categories that we would expect to be changed if Fn14 is important for regulating synapses, also through this chromatin modification mechanism. A question that arose that I think is probably one of the more important ones, and you might be asking yourself this right now, is what was the overlap between the functions or the identities of genes that seem to be regulated by Fn14 at the level of chromatin, and the genes that we identified as misregulated in our single nucleus RNA-seq data set? Well, we did observe about 85 genes that were actually shared by both data sets, which we think serve as like an interesting, potentially higher confidence subset of genes that are likely regulated by Fn14 and likely regulated through this chromatin process. And those genes represented mostly peaks that were opened in the Fn14 knockout and some peaks that were closed. So we can take advantage of the fact that we're fairly confident that these shared genes may represent the most significant, or at least the most reliable targets of Fn14 in a functional way. And we could then perform a motif enrichment analysis, where what we essentially do is we ask at these inducibly changed chromatin regions that the DNA sequences give us any sort of ability to guess what transcriptional regulators might be involved here. So by looking at whether there are any motifs significantly enriched in those peaks versus sort of the background of the active genome, we can ask whether there's any enrichment for particular transcription factor binding sites. And when we did that analysis, we actually did find a couple of really interesting hints towards how Fn14 might actually be mediating these changes. And the first thing that we thought was really exciting is there's a transcription factor motif for a transcription factor that's called FAP1, and it's actually present in about 70% of all of these differentially accessible peaks in the Fn14 knockout, which is really interesting. There's actually not that much known about FAP1, unfortunately. It is known that it's basically a monogenic cause of familial myoclonic epilepsy, which is extremely interesting to us, particularly because epilepsy, it's a disorder of heightened neuronal activity. So it makes sense that there would be some synaptic changes involved as well. But one of the downsides of FAP1 being poorly understood is that there weren't as much tools to actually ask whether Fn14 might be modifying it functionally. On the other hand, we also noted that about 25% of the Fn14 regulated peaks were actually predicted to bind to AP1 factor transcription factors. And AP1 family of transcription factors, as most people know, are really important stimulus-dependent factors that have been particularly implicated in important activity-dependent processes in the brain. I think most people have heard of sort of the most famous family member, FOS, because FOS has actually become a really useful marker of neuronal activity in the brain. And indeed, we see when we look at the sort of expression of Fn14 and FOS together in the DLGN, there's a really high degree of overlap, such that the cells that express a lot of Fn14 also express a lot of FOS. And if you actually think about that, it makes a lot of sense, because I told you that the way we discovered Fn14 was through a transcriptomic screen where we stimulated mice and we looked at what's expressed in the neurons. And as we know, FOS is an activity-dependent gene and Fn14 is an activity-dependent gene, it makes sense that there may be some correlative function there. And so Austin in my lab actually did an ELISA to look at the protein level and see if he could detect any signs that AP1 factors were more or less activated in the absence of Fn14. And what he found is that one parameter of AP1 function, which is the phosphorylation of a family member called Jun-C, or C-Jun, and this phosphorylation event is associated with the activation of this factor. Austin found that there was actually significantly higher phosphorylation of C-Jun in the knockout compared to the wild type. He also found a trend towards an increase in the expression of FOS in general in the knockout as well. And so we think this is really exciting information, and it makes sense that we might see these, activated factors in the knockout, because remember I told you that the majority of attack sequencing peaks that we found to be differentially accessible were actually more accessible in the knockout. So it makes sense that you would have more AP1 function in the knockout, which means if we take a step back, our model would sort of suggest that Fn14 in and of itself is able to shape transcription in chromatin in part by repressing AP1 factor activation. And while not much is known necessarily about the pathways that repress rather than activate AP1, a number of pathways have been thought to do this, including notch signaling. And so we think signaling pathways from microglia to neurons could be an important way to potentially sort of shut off the response of the cell to sensory experience or to neuronal activity. And so in a nutshell, this was really interesting finding for us. And we think we're sort of at the beginning stages of trying to disentangle really a mechanism through which this particular cytokine receptor can link synaptic refinement, because we already know it's critical for that functional aspect of circuit development, but also epigenetic maturation of neurons, which occurs at the same time, but is much more poorly understood. And because, as I told you, Fn14 is actually the receptor of a cytokine that's expressed by microglia, this made us really interested in understanding whether microglia as a cell class could also be sending additional signals to neurons to shape the epigenomic maturation as well. And as I mentioned to you before, mostly we think about microglia as playing very localized roles in synapse elimination. So it would be really exciting to see whether microglia can actually shape neuronal transcription and epigenomics more globally. And so to address that question, we used a pharmacological reagent called plexicon, which we can administer to mice in chow, so it's very easy to use. But this plexicon is a antagonist for a certain cell surface receptor that microglia need for survival. So what happens when you administer mice chow that has this plexicon drug in it, it actually depletes microglia from the brain rather rapidly, actually within 2 to 3 days of administration. And you can see that in these immunostainings here, where I'm just immunostaining for microglia in green. By 3 days post-administration, you can see microglia are largely absent. We're able to keep that state in the brain up through the experiment, which lasted about 8 days. And here's the quantification. So we can very effectively remove these cells specifically during that critical period of experience-dependent. And then we can ask what happens to chromatin. And so what we observed is that similar to the Fn14 knockout mouse, we did find significant changes in the chromatin landscape of the tissue that we were looking at. But sort of dissimilar to Fn14, we found that while in the Fn14 knockout, we found more sites were being opened, whenever we lose microglia, it looks like more sites are actually closed. And specifically, we found about 194 sites that are actually less accessible whenever you get rid of microglia, compared to 48 sites that are more accessible when you get rid of microglia. Now, I think anybody who's potentially familiar with this type of experiment would be quick to point out the fact that we're actually looking at tissue where we've gotten rid of a cell class in the brain, right? So we have to also sort of think about the idea that some of the peaks we're identifying are probably simply missing because we lost microglia. And of course, that's certainly true to some extent, but we were able to filter out a lot of those peaks such that now we can put these differentially accessible peaks into a data set, and it predicts that we're largely looking at neuronal peaks, which makes sense because this experiment is done in the DLGN, and the DLGN is relatively 80 to 85% excitatory neurons. So we think that we're on the right track here. One of the things we're doing in the background is we're gearing up to do this experiment in a more precise way, such that we can actually isolate neurons specifically from mice with or without microglia. But we think that we found some really interesting findings that really motivate that experiment. And so this is, again, just a couple of examples of attack-seek tracks. Here I'm showing you an intron in a gene called synaptoporin, which is really important for synaptic function. And you can see that there's quite a bit lower accessibility of this region when we deplete microglia. We can also see the reverse effect, for example, in this ion channel here, CACNA1G, where you have actually increased accessibility when you lose microglia. So can we take a step away and can we do an ontological analysis to ask in an unbiased manner what types of genes are being regulated by these cues coming from microglia, presumably? And we find a number of interesting categories. One of the things I think is kind of interesting here is the regulation of ERK1 and ERK2 signaling, as this is a really predominant sort of intracellular cascade that in particular is important for activating AP1 transcription and activity-dependent transcription. So it actually mediates communication from the synapse to the nucleus. So I think that's actually a very interesting finding. Of course, we also see just key regulators of neurotransmission, of actin cytoskeletal reorganization as well, which I think is really important because this process of synapse disassembly we're imagining will involve quite a bit of cytoskeletal remodeling. And so a lot of genes that may be really integral to neuronal function and neuronal plasticity may potentially be regulated by signals from microglia at the chromatin level. And if these changes that we find in chromatin organization in the absence of microglia are relevant to synaptic refinement, then we might observe that if we remove microglia from the DLGN during that critical period P20 to P30, we would observe a deficit in synapse refinement or synapse elimination. And indeed, that's exactly what we see. So whenever we use plexicon to deplete microglia, the same paradigm I described earlier, and we look at dendritic spines in the DLGN, which we previously showed to be a really nice sort of structural correlative functional refinement, we observe that there are significantly more spines in the DLGN when we deplete microglia, whether we're looking at total spines or whether we're looking at a couple of different subsets like mature spines or immature spines. And so this is really consistent with microglia contributing to synaptic refinement during this experience-dependent phase in the brain. And so, all of these data sort of gave us the sense that Tweak and Fn14 and microglia in general are doing some really important things at the level of epigenetic regulation and transcriptional regulation in neurons. This made us interested to kind of take a step back and to ask, is this something that's relevant beyond the visual system? As I mentioned, I really love the visual system as a model of experience-dependent development. But actually, one of the drawbacks, it is actually kind of difficult to assess visual function in mice at the behavioral level, compared to other behavioral assays that may be more amenable to the analysis of Fn14. And so we wanted to ask whether Fn14 was expressed outside of the DLGN, and if so, where is it expressed? And so we looked at Fn14 expression across the brain, and I'm just showing you an example of a sagittal full mouse brain at P27, the peak of refinement, and I think you can really nicely see Fn14 expression in white across a number of brain structures here. So for example, we see really high expression of Fn14 in the cerebellum and the granule cell layer. We also confirm that we see high expression of Fn14 in the DLGN here in this pink box. But we also see Fn14 expression in the hippocampus. Looks like it's in sort of select populations in the hippocampus, which is very interesting to us. And when we look at the when we look at the overlay of like FOS and Fn14 in the hippocampus, particularly the dentate gyrus, we again see that there's quite a substantial overlap between these two markers. And we became interested in whether Fn14 might also be regulating synaptic development and function in the hippocampus, which is a brain region that's very important for a variety of functions, including learning and memory, for which there are much more tractable and quantitative assays for looking at circuit function. And the other reason why we thought it would be really interesting to move towards the hippocampus as a model, or at least as a way to think about the larger scale functions of these molecules, is the recent publication of a paper by collaborators of ours where they were actually looking at hippocampal neurotransmission and asking how manipulating tweak Fn14 signaling impacts that process. And what they observed is that at baseline, neurotransmission is sort of dampened when you wash on tweak. So when you put in tweak to activate Fn14, there's a dampening of synaptic neurotransmission, as well as long-term potentiation, which is something that's really important for memory, and which is something that requires both gene transcription as well as structural and functional changes at synapses. So we thought that this could potentially give us a platform for, in a behavioral way, asking what is Fn14 doing in the intact brain? And so we looked at that by using a couple of different memory analyses, and one of them is cued fear conditioning, which I'm sure many behaviorists in the audience are familiar with, but I'm just going to briefly describe this assay. So what we do is we put mice in a cage that has some context. In this case, it's stripes across the cage. And then we provide the mouse with a sound or a tone along with an aversive foot shock that the mouse does not like. And so the mouse becomes afraid of this context and of this tone because they remind it of the foot shock, and it exhibits that fear by freezing. So you can then take the mice out of that cage, You can put them into a cage where the context is the same, but they don't receive the tone. And you'll notice that they observe increased freezing because they remember that connection. You can put them in a novel context and they actually won't associate it with the shock. So they won't freeze that much. They're not just freezing all over the place. They're freezing specifically because they remember that association. So what we observed in the Fn14 knockout mouse compared to wild type littermates was that the mice learned to associate the foot shock with the context and the tone perfectly well. That's why they froze roughly the same amount during the training phase. But when we re-expose the mouse to the same context without the tone, we noticed that Fn14 knockout mice tend toward freezing less than wild type littermates. This wasn't an overall problem because when we put the mice into a novel context, they froze the same amount as wild types. But we noticed a significant decrease in the amount of freezing when we re-exposed the animals to the tone, but not the context, which indicates that Fn14 knockout mice tend to learn normally, but they show deficits in memory. We next asked what would happen if we removed microglia and did a similar experiment. And what we observed was that while we didn't see as robust of changes as we did in the Fn14 knockout, we did observe a relatively strong trend toward a decrease in freezing when the mice were exposed to the tone in a novel context. That's exactly sort of the feature that we observed Fn14 knockout mice showing the greatest deficit in. And so we think that's kind of promising, that there could be some correlated role for these factors in regulating that process. But my postdoctoral fellow, Austin, was also curious about, looking at a task that maybe was more hippocampal dependent, because this fear conditioning task can involve a lot of different brain regions. And so he next subjected the knockout mice to what's called a Morris-Water maze, which is another just really classical assay of hippocampal memory function. And so what we observed is that the Fn14 knockout mice were perfectly capable of finding a hidden platform during the training phase. Actually, it's not hidden during the training phase. But when we removed the platform, or when we made the sort of water opaque, we noticed that Fn14 knockout mice swam significantly less in the quadrant of the pool where the platform had initially been. That suggests that the wild type mice are better able to remember the location of the platform once it's removed compared to the knockout. So this again hints that Fn14 may be critical for memory function. And given sort of our hypothesis or our data, which I think suggests that Fn14 may actually be playing an important role in coordinating the transcriptomic and epigenomic profiles of neurons, and that those processes are important for the roles of Fn14 in the brain, we also wanted to ask, in combination with the evidence that Fn14 is very broadly expressed in the brain, we also wanted to ask whether there were any sort of large-scale changes in neural circuit activity. And so we looked at that using EEGs, where we were able to implant an EEG probe towards the posterior region of the brain. So if you notice this red dot here, it's actually quite close to those structures of visual cortex, DLGN, and hippocampus. So it makes sense to put the probe there. And what we did was we implanted the EEGs. We allowed the mice to heal. We then did EEG recordings over 7 days, just normal baseline. And then what we did at 9 days is we actually injected the mice with a neural convulsant called PTZ, which basically inhibits GABAergic transmission, which is inhibitory. And we asked how the mice responded to this convulsant. And what we observed was that at baseline, all of the different types of neural activity were relatively normal in the Fn14 knockout. However, when we induced seizures in the knockout, or when we actually injected a convulsant into the knockout, we observed that Fn14 knockout mice were significantly more susceptible to seizures and to the deleterious effects of seizures. So for example, these are 2 example traces of what we observed in the EEG recordings of a wild type mouse after injection with PTZ versus a knockout. So in the wild type, we injected here at this black arrow, and eventually we came to a general tonic-clonic seizure. And that's what this looks like here. And the seizure would happen, and then the circuit would go relatively back to normal again. What we noticed in the knockout was that these seizures happened much more quickly. The seizures themselves were sort of longer duration, and that the mice were much less likely to survive these seizures compared to wild types. So I'm just showing you some of those quantifications here. where we have the number of mice with general seizures is increased when there's knocked out Fn14. We also see the latency to seizure is decreased. We see the duration of the seizures is increased. We see that the mice survive less when they don't have Fn14. That's what I'm showing here. And we see that overall the fates of the mice after this sort of manipulation are worse in the Fn14 knockout. And we think that that's really sort of interesting and consistent with what we know about Fn14 function, actually, because we know that in the Fn14 knockout, we have too many synapses. These synapses aren't appropriately strengthened, right? So we already have too many synapses. And you can imagine that if you have too many synapses and you give additional excitation, you could actually go overboard in terms of excitotoxicity. The other reason why I think this is kind of interesting in light of the chromatin accessibility data is that the synaptic genes that are seeming to be regulated by that process would be genes that we might predict would be able to potentially downscale the number of synapses, which is an important part of a healthy brain's response to seizures or seizure-like activity. So we think that the inability to sort of structurally or plastically remodel chromatin and transcription may contribute to these phenotypes in the knockout mouse. And so we've been able to develop a model for how we think this process might be working. I want to highlight that this is still a sort of speculative model. We're still sort of trying to disentangle the mechanisms, but this is what we think might be happening so far. So here I'm showing you what we think is going on in the nucleus of a neuron during experience-dependent refinement. I'm showing you here some histones with these little tails that can be modified. I'm showing you E represents a cis-regulatory region. We call them enhancers. And then G represents a gene that enhancer regulates. These little pink circles represent modifications. So you can consider these to be, say, methyl marks or acetylation marks. So what I hope you can appreciate here is sort of steady state. We have a number of genes that are accessible but are not being transcribed. And then we have a gene that's being transcribed, even though its cis-regulatory element is not accessible. And then we have a gene that's not being transcribed. Whenever we have Fn14 turning on in the neuron, which happens in response to visual experience, specifically during this critical period of development, we speculate that this allows certain machinery to bind to these cis-regulatory elements, and that drives the expression of a couple of different types of genes. Synaptic genes, which I told you are many of them vastly downregulated in the knockout, as well as modifiers of chromatin and histones, HDACs and KDMs to be specific, or histone lysine demethylases. These genes go to the synapse to start to remodel or disassemble them. Sorry, the protein products of those genes go to the synapses. The protein products of these actually come down and modify the genome further. So now we have these two genes that were being expressed or not expressed here. Now with the HDAC KDM function here, we can see that this enhancer becomes available, but is maybe bound by a transcriptional repressor to turn it off, while this receptor becomes accessible, and now that gene can be expressed. And we think that this is working to essentially allow tweak Fn14 signaling at the synapse to provide feedback to the nucleus to drive these genetic changes, which come back and regulate synaptic plasticity, including learning and memory and excitatory tone, which is important for the seizure susceptibility phenotype. But additionally, we see that microglia play a function in this process as well, probably largely through pathways that are non-Fn14 dependent. While we did see some limited overlap between some of the genes that are being regulated by microglia and Fn14, the majority were non-overlapping. So we think that microglia probably employ a much broader repertoire of molecular mechanisms to sort of shape neuronal transcription and chromatin. And so we're really looking forward to following up on that result soon. And so essentially just want to end with the acknowledgements. Thank you very much for your attention today. I want to thank everybody in my lab. Johan is a research technician who was really instrumental in the attack-seek data analysis. Uma played a large role in helping us to validate some of the gene expression changes we saw. Austin was, of course, the postdoc who really drove the project. And everybody else in lab really has provided fantastic input on the paper. And of course, want to thank our funding sources. And with that, I'll be happy to take any questions.
Great. Thank you, Lucas. So questions, you can raise your hand, you can throw something in the chat if you want me to ask the question. I know that Nirupa was the first up, so I think she can have the floor.
Okay, Lucas, what a terrific talk. Thank you so much. This was really nice. Can you hear me?
Yes, I can.
Oh, okay. So you got through a lot, and I have to compliment you. You're an excellent teacher.
Thank you.
So I think that people who aren't familiar with a lot of the technology probably at least got the outline. So that was very nice. I have a technical question, which I'll save for after some other people go. But I want to come back to the experience dependence part of it, which may kind of tie in with the learning and memory thing. But what kinds of mechanisms are you seeing that that activity of RGCs impinging on these neurons in the LGN and the DLGN? What is that doing to activate Fn14? Because that's sort of like your starting point, right? So I know that it was one of your top targets in the differential single-cell RNA-seq that you looked at.
Absolutely, yeah. Yeah, so I can certainly speculate on that. So the lab that I was in as a postdoc, Michael Greenberg's sort of focus is on understanding immediate early genes. And yeah, so you're clearly aware of this. So one of Michael's favorite topics is AP1 transcription factors, FOS, INPAS4. INPAS4 is not an AP1, but you get my drift there. So the way that we think about experience-dependent or activity-dependent transcription is working is that there's a really quick response where you turn on transcription factors like FOS that can then bind the genome at particular locations to drive the expression of what we call late response genes, which turn on around 3 to 8 hours after that initial sort of transcription factor binding. We think it's very likely that Fn14 is a target of one of these transcription factors. We don't know which one yet, but I think FOS is a very promising, FOS and other AP1s is a very promising possibility. And the reason we think that is because we did a couple of experiments early on where we actually happened to have like FOS knockout fibroblasts. And because Fn14 is really highly expressed in non-neural tissues, we asked whether FOS is important for the stimulus-dependent induction of Fn14. And we know that the induction was dampened by at least 50% when we got rid of FOS. So that's one of the reasons. I also looked in the FOS knockout DLGN of just a constitutive knockout and observed that Fn14 expression was lower. So I think there's a high likelihood that AP1 factors can induce Fn14 expression, which I think is really interesting in light of our result that Fn14 may then come back and provide some feedback onto those pathways.
So in the FOS global knockout, then you see the LGN projection neurons not maturing?
So we didn't look at the maturation of the LGN projection neurons in that model. I'm actually very interested in this because what I didn't talk about today is part of our lab actually is studying all of these genomic changes, but in microglia. And we've done some experiments in visual cortex where we look at stimulus-dependent ATAC-seq in microglia, and we find AP1 factors are just blowing up. Like that's what coordinates those responses. So I think that's really interesting as well. But yeah, I would love to look at that. It's a little hard to study the function of one particular AP1 factor because there's so much redundancy. It's a large family of TFs, they dimerize. So one of the models we were using in the Greenberg lab was a triple floxed conditional knockout, FOS-B, Jun-B. And I'm trying to get my hands on those. I'd love to look at that. And I would expect to see, a similar phenotype in refinement like we see in the Fn14 knockout.
Okay, great, thanks.
Other questions for Lucas? Just chime in. Europe is back. So, I had one question actually. So, oh, go for it.
Oh,
go on. Yep.
Sorry. So your main hypothesis is that the synaptic refinement is mediated by an interaction of Tweak and Fn14. But some of the other activities might be independent of Tweak, no?
They definitely
So is there any, I don't know, I don't know this, but is there available a Tweak knockout model in which you could sort of, or use inhibitors of Tweak, specific inhibitors to sort of dissect those that are Tweak dependent and Tweak independent, and that would also lead perhaps to the dependence on microglia of all of these activities.
Yeah, for
because
sure.
The microglia depletions are kind of too pleiotropic treatment, not so it's a very drastic thing. But if you could just go particularly to Tweak, that might be a better.
Yeah, we did that experiment, and that is in the paper. I just didn't have time to talk about it today. So when we look in a constitutive Tweak knockout mouse, we do see transcriptional changes in the thalamocortical relays of the DLGN. Those changes are largely non-overlapping with the changes that we see in the Fn14 knockout. First of all, there are many fewer changes, which I think makes a lot of sense because Fn14 is the neuronal receptor. Additionally, Fn14 is much more highly expressed. While Tweak turns on in a subset of microglia, Fn14 basically turns way up in all neurons. So we think that Tweak is maybe a more subtle or nuanced sort of regulator of genes. And consistent with that, we find that there are probably around 100 genes that are misregulated in the thalamocortical neurons of Tweak knockout mice. And these are non-overlapping with the Fn14 targets for the most part. But what's interesting about them is they're still encoding regulators of synaptic development, as well as chromatin assembly and disassembly. So they regulate a factor called SET, which is important for regulating methylation. And they also regulate members of a transcriptional complex called the SWI-SNF complex, which have been very heavily implicated in neurodevelopmental disorders. So kind of what you described, what we think we're uncovering here is that our Fn14 knockout data set may be sort of enriched for targets of Fn14 that do not require Tweak, whereas when we remove Tweak, we're able to uncover some of those targets that are specifically mediated by Tweak. And I completely agree that the plexicon experiment is certainly somewhat of a sledgehammer. It's really a discovery tool to get us started. But we're hoping it might, enable us to sort of uncover some things that we can then go and test in a more precise manner.
Could I ask my technical question while somebody else? In your ATAC-seq, I don't know if you said this and I just missed it, but had you filtered out all of the interneurons and the glia and everything else? It seemed like there just weren't that many different neuron types. So like there should be a whole lot of interneurons right there.
There aren't that.
Well, there's a bunch of GABAergics, right? And
well,
then
here, I'll say this. One of the sort of weaknesses of those experiments that we're now sort of working on addressing is that we're not specifically looking at a particular cell type in those experiments.
right.
So yeah, so we're looking at a heterogeneous.
Well, you can't with the ATAC.
So we're looking at a heterogeneous tissue that's largely enriched for neurons. And particularly in the plexicon data set, we did a lot of filtering to try to get rid of some of those microglial genes. What we're doing now is we're going back in and we are using an approach that we already use for other reasons called SUN1 mice. This is a nuclear tag that's CRE-dependent. So what we can do is we can go in and specifically purify the populations we care about. And now we can ask in a more precise manner what's happening at the level of the genome. So those are the experiments that we're ramping up to do. And I think we'll have a lot more cell type specificity there.
Right.
That's actually the approach we use for microglia, that we use the SUN1 approach and it works fantastically for isolating microglia out of the brain. So.
Okay, great, thanks.
Mm-hmm.
Okay, well, thank you so much.
Thank you.
Thank you.
Talk to you soon, bye.
Bye.